So sorry everyone for the slight delay. We had some issues with the sharing of the speaker screen. So in the end, I will be sharing my screen with the presentation, and Benedikt is going to tell me when to change slide. So let me start with a brief introduction on who Benedikt is. He's a PhD candidate at EPFL. He's working at the interface of computational biology and protein engineering. Before his PhD, he did an internship in the University of Washington in Baker's lab. And his research focuses on developing advanced computational tools and pipelines to enable and accelerate protein design. Today, he's going to introduce a simple pipeline, computational pipeline on how to proceed with protein design and how to score different designs for later experimental evaluation. Benedikt, the floor is yours.
Thank you so much, Davide. So yeah, as also from my side, a very big sorry for the delay in sharing the screen. I was challenged with the Google Meet tools. So one should say that one is capable of handling these different presentation tools, even though you work on like developing deep learning models, but unfortunately this is not the case. So yeah, as Davide already mentioned, I'm working in the lab of biomolecular modeling. Next slide, please. Where the lab is headed by Matteo Dal Prado, and we mostly work on a lot of different things. So we have also a group which works on molecular assemblies or membrane proteins which are in the membrane, but we also have a group which works on nanopores. But there's one small section which I'm part of, which does protein design, and we are located on the beautiful La Clément. Next slide, please. As you can see here, this is the EPFL campus, and yes, it is really this pretty in person, so I can certainly recommend to visit us at some point. But yeah, this is just a short introduction of where we are and what we're up to. Next slide, please. And I just want to get a brief overview on like what is protein design, what has it been, what is it now, and most of the figures I took here are actually from when the Nobel Prize was awarded 2024. And this is just a short overview for you to get on the same page. And I think some of you attended already Ayasia's talks, where he introduced Cypred to you. And to me, this is also something which is more part of the earlier methods. But now with the advent of deep learning, and especially also with like many more methods coming from different labs, or in the end, not that many different labs, the world of protein design has really changed a lot. So back then, and this is also something which I talk from hearing from others, I've not done it myself, fortunately, the protein design process was very tedious. So you would have to really do heuristic-based design. So you would a lot of times actually print out your protein and model it in a three-dimensional paper structure. You would follow certain rules on after how many amino acids you would now be allowed to do a turn, what certain handedness for the sequence of the residues actually determines what motif you're making. There were a lot of heuristic-based formulas which were not really of that much physical origin, which kind of yielded them more working designs, sometimes less, sometimes more. And then assigning the sequence, like bear in mind you only had the protein backbone and nothing else, was very complicated and tedious. So a lot of times you would just look at it and be like, this alpha helix, like on the slides here, it's touching the beta sheet, it should be more hydrophobic, and this one is more exposed, so it should be a bit more like hydrophilic, so maybe this works, maybe not. And then you would use this as an input into the software called Rosetta, which is a huge software consortium which was founded by David Baker when he decided that protein design is such a complicated task that we need to have all hands on deck and not just one single lab doing it. And this software is still being used, and it's still perfectly fine at what it does. But that specific task was very, very slow, and it took a long time to assign these residues to the backbone. And hundreds of thousands of CPU hours were used and wasted. And this protein here, you can see here in green is top 7, one of the most famous proteins ever de novo designed, and this is also why it was showcased by the Swedish Academy of Sciences. So what is protein design now? Next slide, please. And in this case, you really have to bear in mind that the field of deep learning itself is also a quite new field. Like when you think of the attention is all you need paper from 2017, where the transformer architecture as we know it was first introduced, it kind of also didn't take that long to go into the field in protein design and revolutionize a lot of things for us as well. And this is also why it's very important to mention AlphaFold. And I'm pretty sure all of you have an idea what AlphaFold is, but I'm just very, very briefly mentioning some caveats or some things I like a lot about it. And most of them, well, they're details, but they make it very interesting. So we know it's a structure prediction network. It's trained on the whole PDB. It will give you the structure of a sequence with some evolutionary information. So for example, in the figure you see down below, there is this pairing input, which are the so-called templates. And this is something that Cypred actually used. So this just as a kind of a bridge from what you heard from Ayasia, was used in order to generate these two-dimensional templates from the MSA. So this was an additional representation of the protein used as an input to the AlphaFold network. But what AlphaFold 2 did differently, next slide please, was that instead of just having the MSA as an input for the network, it uses some structural operations called invariant point attention, where it is allowed to learn importance over individual frames consisting of the C alpha, the C beta, and the nitrogen, but also something which is called self-distillation, where the model actually predicted a lot of structures from UniProt, so there were no structures available, only the sequences from there, and then it used its own confidence to then filter these structures and train on those again. So there are actually some advances in the field of deep learning through the network of AlphaFold, which is also why this network was awarded with the Nobel Prize. That's why Demis Hassabis and John Jumper shared the prize there. And this is just for me the pinnacle of how deep learning helped advance things in protein design. So next slide, please. We have there's a bunch of abbreviations here. Some of them might be known, but we have, for example, the so-called denoising diffusion predictive models, or DTPMs, where you would sample from some sort of distribution to then go to something which is sensical and produces something which is part of your data, part of the whole generative model part. And in there, you for sure already have used DALL-E or NanoBanana or any of these image generation tools, where you would then go ahead and prompt the text and say, oh, please, I want a cowboy riding a horse with an astronaut suit or something. And then you have this information in there. But now instead of this conditioning of your textual input, we would condition on a protein which is in nature. So this is this nicely rotating black structure on the right in the slide here. And this would be the conditional input. And then based on that, these models, and specifically here RF diffusion, would go ahead and sample from a three-dimensional Gaussian of coordinates and just pick these single residues in a trajectory, so that in the end you will get this protein binding your new protein as a conditional input. So this is something which was very, very revolutionary at the time. It was actually not the Baker Lab which came up with this first. There were other tools which came with it, but they sat on top of their own AlphaFold version, RosettaFold, which was very, very successful to then put everything all together and have this structural generation tool. There's also the field of the message passing neural networks, or graph neural networks in general, where message passing neural networks are one sub part of it, where when you think of a protein, like you think of the protein backbone and the residues as a graph, which is some sort of mathematical abstraction of something with edges and something with nodes. So for somebody with a chemical or biochemical background, it makes a lot of sense to inherently think of a protein like this, because your atoms are the nodes and the connections between the atoms, the bonds, are the edges. So that makes perfect sense. So if we were to abstract our protein backbone in this graph-like structure, we can suddenly use a lot of these very cool graph mathematics, which very smart people from the field of machine learning came up with, and we can use them. And we can train models on this, and we can, for example, do the sequence reassignment task with one of these networks. because it makes sense that one residue in the graph structure only thinks of one residue close to each other, because that makes much more sense than something which is much further away. This is where these graph neural networks are insanely efficient and very useful. And then there's, of course, the large language models, right? Also some sort of transformer, as we all know at this point. But in this case, we actually forget the protein depth structure. Yeah, I know this is crazy, but it actually works pretty well. Like, proteins can be abstracted as a language. And there is one very famous one, which is called ESM from Meta, where they essentially abstract the protein sequence, and then they only train on masking. So they would take the whole protein sequence and they mask out one single residue in there, or a second or a third, and let the model refill this gap based on the patterns it has learned from the data of everything. And it turns out that it does pretty well in this. And it also turns out that this can be used for reflecting very, very, very subtle evolutionary signals in very big protein alignments. And it can also be used to break the structure, because we know that sequence leads to structure. So these three networks, or these three, I would say, different hallmarks of deep learning have really went along together with protein design. And I will then not mention these architectures anymore, but I will do a lot of name dropping of what models we use these days. But I will maybe mention one does one thing or one does the other thing, but in the end, they all can be traced back to these architectures, except maybe for some very few other ones. Okay, next slide, please, Davida. So now, what is protein design now? Instead of any protein, which is this conditional target input, it could be a small molecule or a nucleic acid. Next slide, please. We would go ahead and we would design the backbone of the binder. In this case, the protein in red. As I said, it could also, our target could also be a small molecule or nucleic acid type. And we would mostly go ahead and use RF diffusion 3 because it's open source, it's available, it's very easy to use, but there's also alpha protein and chroma. Next slide, please. Then we would go ahead and design the binder sequence. For this, we would use protein MPNN or ligand MPNN, but there's also tools like ESM inverse folding or Carbonara. Next slide, please. Which conveniently has been developed by our lab. So this is a little teaser, a little advertisement here. It's not so well known as protein MPNN, but in the end, it's also a graph transform, which works pretty well. Next slide, please. And then we would use a feature, and this is something where I previously mentioned the confidence. From all these forward folding methods, such as AlphaFold 2 or 3, Rosetta Fold, ESM, Chai, Bolt, they have to give out a confidence on how confident they are with the structure they predicted from the sequence. And we would then be able to use this confidence and link it with the success in the lab. And without this, we would not be able to design so many sequences, because if you sample from a three-dimensional Gaussian, you inherently introduce noise, because it's a stochastic process. You sample from a lot of different parts, and in the end, if you also generate sequences to these backbones, you would then get a lot of other different sequences. So it's a very, very noisy process, and in order to go down to the wet lab and really spend money or put hands on deck, essentially, you want to be quite confident and that the designs you made actually do work. And for this case, in this case, we were very happy. And like when RF diffusion came out, they also mentioned this, that AlphaFold 2 was very, very important in order to help them filter that design. So there are certain metrics such as the PIE or the PLDVT or other different metrics which now come out, which can correlate somehow weakly with if this value is high or low enough, this design might actually yield something in the lab. Next slide, please. And then we obviously have the very important in vitro evaluation, because this talk is supposed to be also about the wet lab part. So just as a little disclaimer, I mostly work on the dry lab things. I have had a training in the wet lab, but it's been a while that I actually applied it myself. But obviously, in order to test if we have working proteins, we need to express them, we need to purify them, or sometimes purify them. And then we need to do all sorts of very nice biochemical assays, biochemical analysis methods, from CT spectroscopy to melting curves to affinity measurements, and best case scenario, maybe even solve the structure using cryo-EM or XRD. Next slide, please. So there's also other tools right now which are actually capable of skipping all of these steps, these pipelines where you run one software package after the other, where you just say, oh, I have this target, and now I would like to go to the in vitro evaluation directly. I don't want to bother about these details in there. I just want to design proteins. And one of those are, for example, Bindcraft and BoltzGen. And next slide, please. Bindcraft will be presented by Leonard, I think on next Thursday, if I'm correct. So this is just a bit of a teaser on what is yet to come. And for the in vitro evaluation, next slide, please.
There is a small error in the poster there. The event is correct on the Luma page. It will be next Friday. But yeah, right. Sorry for that.
No worries. So for because I mentioned how cool it is to design proteins, right? I mean, it's obviously really cool that we can make something completely new to nature and novel, but it's still hard to test those things. So almost on par with the RF diffusion, there was another method which came out of the Baker lab, which is called semi-automated protein production, or SEP. And if you talk with people from this lab, they will call it cowboy biochemistry, because it's so fast that it shoots from the hip. I didn't come up with this, but it's kind of a bit of the running gag there. And the idea is that after you've designed your sequences, you're quite confident with your designs, you would usually go for something called an 196 well plate, which is decently expensive, but it's not so bad. And the way it's done is that, like, after the gene synthesis, you go to GeneScript or to Twist, and you order your genes, your gene fragments. You would then use Golden Gate assembly to put these into vectors. And from there, it will take around 48 hours to actually get valid results. And the cool thing here is that the purification will also give you the first information. So if you use this SCC in step 5, the SCC purification, if you see that the trace is not well behaved, you see that it's not monomeric, for example, this already gives you a first kind of criteria whether or not your design has worked or not worked. And it can be done high throughput. And it does say here that it's approximately $5 per construct, assuming that you already bought all the automated lab equipment like this robotic pooling or the HPC for the purification and all these other very not so cheap designs in the beginning. So I think the robots for the pooling, I think this is like, I don't know, 300 to 400K. So it's not so crazy cheap. But I'm also very happy to give this knowledge here further to other people who will then talk about it in a bit more detail, because this is just my very rough overview and, well, maybe also the closest I will ever get to this. All right, next slide, please. This is just one real-world example from our lab, which actually Davide, who is also in this meeting, did himself. And it was essentially a way to test whether or not the enzyme we designed does what it's supposed to do. And you can see, well, some of them transition from yellow to red, some of them actually do work. And you can also see that you can also get different concentrations. I mean, it's a biochemical assay. I don't think I need to explain what it does. It's just that it actually is tractable. You can very easily then go from the dry lab to the wet lab, whilst knowing that you have a system you can work with. Okay, next slide, please. So what can we actually now do with it? And this is actually the part which, when I talk about this, or when I have this presentation, I enjoy the most, because usually you biologists, synthetic biologists, who know your problem much better than anyone else, have the best ideas for this. Because I only know these methods, I only know how to develop them. I can help you set up things on the cluster. I can try to give you opinions on how to do enzyme design. But in the end, I don't have a specific biological problem. Usually I get approached by people who have this, and this is something which I enjoy a lot. But just to give you a showcase what has been done before, and this is definitely not up to date, but more cherry-picked on what I liked. Next slide, please. So this is just a huge summary of different proteins where people just try to make proteins with functions. They did protein binders. They did certain nanocages. They tried to follow certain topologies. They tried to make specific oligomers, sometimes even enzymes, some of them in a more or less interesting fold state. So this just gives you a bit of the wealth or the vastness of what protein design has done. And I think this paper is already 3 years old, so I'm pretty sure, like, if we were to get all the de novo design proteins, they would not fit on one slide, and also probably not on 10. Next slide, please. But here you can see, like, people try to sculpt nanopores, which are de novo designed, like some transmembrane beta barrels. Next slide, please. Or they try to design binders against these intrinsically disordered snake venom toxins, which can be used to, like, super fast treating. There's antibody design with using a fine-tuned version of this RF diffusion I already mentioned before. Next slide, please. Which actually Nate is going to talk about. next Tuesday, so it's definitely worth to tune in. It's a really cool paper. And then next slide, please. There's also obviously people who, like I was already mentioned, put all of those things together in one pipeline. And then next slide, please. There's the thing which I'm mostly interested in. Can we actually design enzymes using these tools? Next slide, please. So yeah, you can clearly tell that from the beginning on of protein design, things have moved further and further, and this is also why David got the very, very deserved Nobel Prize for this. So this is all available on the webpage of the Nobel Prize, but I can certainly recommend just to give you an idea of what people did, but also like what is possible. Next slide, please. Yeah, are we done? Like kind of, we can make new proteins, right? We can do whatever we want. It's fine, no? Why do I bother researching at this? It's finished. Well, no, not so much. So there's still a few open questions and still some challenges to solve, and this is more to give you an idea where the field is currently at. Next slide, please. There's obviously the reflection of dynamics, and this is something where progress has happened a lot over the last 1 or 2 years. There's people who try and incorporate molecular dynamics data. There's people who try to specifically work with NMR data in the PDB. There's also just people who try to find this inspiration from physics-inspired neural networks and see if this can actually be used to correctly reflect the dynamics of proteins. So I linked one of these papers there, but like this is something which has been developing further and further. Next slide, please. But there's also obviously the novel enzyme design, where so far people have mostly scaffolded active sites of enzymes and then built a protein around it. But still, there's some ideas and some approaches flowing around, so maybe it will be a solved task in 1 or 2 years, at least for the simple enzymes, the enzymatic systems. Next slide, please. And then there's the big question of immunogenicity and biodistribution. So whenever somebody approaches to you and says, oh, look, I can design a binder against this really, really bad cancer target, whatever it is, and you're like, oh, that's cool. So you solve cancer, right? Well, unfortunately not. The problem is that if you would use one of these de novo design binders and go to any sort of in vivo version, any sort of in vivo model to it, be it a mouse or anything bigger, there is usually a big problem with your immunogenicity, that these models then usually have a pretty high reaction from the immune system. So the challenge is actually that you will have to design proteins which resemble the human proteins in order to use them as a drug. And this is where the field of antibody design, so definitely tune into Nate's talk, is something which is growing a lot, because you can just take the humanized stem of the antibody and redesign the CDR for a certain function. But then you still have to solve the whole biodistribution issue, essentially the same thing like small molecules. So in the field of, I would say, drug development, I'm not saying protein design is like we cannot do anything, but it certainly is more challenging because we are competing against small molecule drugs, which, as we know, are the workhorse of pharma. So there is certainly other things which might be easier, and there's also like this intermediate thing where we go to peptides, where we can exploit both of the properties, and you could use some non-canonical amino acids to make them more stable. But it still requires a bit more hands-on, and this is a bit reassuring. It would have been too easy out of that, right? So it's good that it's complicated. And then one of the other challenges, next slide please, is that we still are not so confident with these metrics. So I told you that, when AlphaFold 2 says, or AlphaFold 3 says, oh, look, this is a well-folded structure. It has a PAE of, well, the number I wanted to have. Sometimes it also just doesn't work in the lab. And you're like, well, I had this really good design, and it was supposed to work according to all the tools, the prediction tools I used, and yet it just doesn't work. And then there's always this question when we use this big 96-well protein expression and purification: is it because of that we just throw away things which actually would be fine in different conditions and different media, or is it just that these metrics are not working that well? So there's also a lot of optimization going on, active learning approaches, different trajectories of different labs, and this is also where I'm pretty sure Adaptive will tell you something interesting, if not a picture from them here. Because they, as a company, kind of set out to solve these problems because they just gather so much data from all the different customers who approach them. And next slide, please. So now I talked a lot about what protein design is and what you could do with it and what people have done with it and why it is so impressive. But how do you actually do it? Like hands-on, like you have a keyboard in front of you, you have a laptop. How does it work? So it turns out it's actually not so complicated. Yes, next slide, please. For me, there's 3 different levels of expertise and also patience I would require you to put into this. And the easiest one is obviously online. And at this point, there's so many different tools and methodologies who are really easy to use. There's a GUI, it works in your web browser. You can very easily just click around. You don't really have to understand anything about deep learning. Maybe you shouldn't have an idea what a protein is and a protein sequence, but I presume this is very much given here. And it works. Like for prototyping, it's very good. The only downside is if you were to go for a production scale job and say like, oh, I have this hard target, I want to design 10,000 binders against it, this will probably not work. Because you will need a lot more GPU power for this, and it will also just take really long. I mean, if you have a Google Colab Pro account, it might work, but it will definitely take some time. There's also something where a lot of companies who have developed their own proprietary models, like for example, Latent Labs, which is a spin-out out of DeepMind, which kind of have a teaser of their model they sell to companies, and then you could use those in order to just play around with it and get an idea of it. But obviously for academic purposes, this is a bit useless. Then there's the second tier. Next slide, please. Where I'm working on something personally. This is just a work in progress where you kind of have the same sort of ease of deployability, but you would have to install it on a workstation. So if you have a gaming PC standing around, some sort of desktop, you have a GPU on there, you have some static memory, you would be able to run this through a Jupyter notebook. You would click yourself through the different cells, and then you would get the analysis there. So it's kind of this in-between solution where you need to have some resources, but you don't need to go on the last tier just yet. So next slide, please. And this is for me clearly the biggest one, because I mean, most of us, I assume, have some sort of an academic background, and in there is some sort of high-performance computer. So you have an HPC on your local university, or you're associated to some institute, and in there you have very much the possibility to go in a production scale job to produce as many proteins as you want, and then go on there and do the filtering using AlphaFold, and it doesn't matter that much. And I mean, obviously, it's very hands-off. However, you have to have an idea what you're doing. So there's the knowledge to know about the command line, about Slurm, how to manage the cluster, all those things. They are kind of required to go on there. And ideally, the software you're using is packaged in containers. And I remember in the lab where I did my other master thesis, it was a big deal when AlphaFold 2 came out. So not the big lab. And in there, suddenly everybody was like, but how do we actually install it? How do we run AlphaFold on a cluster? And this was quite a big challenge because, well, there were a bunch of very, very motivated biologists and some computer scientists who were like, why do we care? So we had to find some sort of solution which is easier. And this is why I also went ahead, and some of the tools I presented to you already and I'm going to talk about a bit more. They are indeed packaged in containers. If you don't know what a container is, it's essentially an operating system in a box, and it makes running the software very, very easy. So there's just once you get the container binary and then you put it on your cluster, you run it there, and it's easier for you, and your sysadmin will allow you for that. So this is easy. Okay, next slide, please. So what is available online? As a consequence of this meeting, I have actually made a GitHub repository, which I've linked here. So it's here, the QR code also links there. And in there, I've collected all the links, which could be interesting for you to try. So on there is a lot of different tools, from the direct hands-on explanation I showed you before, but also just to play around with it and get an idea. Most of them are hosted on the Hugging Face, which personally I like the most because it's like very democratic. Everybody can push their models, you can give feedback. There's Google Colab, which is a bit more annoying when it comes to updating the models, because sometimes they will just stop working because they changed something on their end and they didn't notify you about it. And since not so long ago, there is something called from NVIDIA, I forgot the name of it, but they also, like it's in the links, but they have a lot of very cool models as well. They host themselves, and since they have like infinite money and infinite GPU resources, it's very convenient that they give something to us poor academics. So yeah, I mean, maybe we can go through those, and if you have some questions about it afterwards, that would be a very good resource to just get started. Next slide, please. So what is available for the cluster? Like, you are working in a university, you're attending this talk, and afterwards you're like, I want to get going. I want to start my own protein design run. Well, first of all, I would recommend you to go for Bindcraft, the Boltzgen. Maybe wait for Leonard's talk. Maybe he has some insights, which I don't have. And then I also went ahead, and in this repository where you can see the links, there's also a bunch of containers. There is the files, how to build them. So they're the so-called definition files or the Docker files. And then B, there's the commands how to build these containers and also how to correctly run them. Unfortunately, I've not found a very easy way to share a lot of binaries with you. So those in total would sum up to maybe 100 gigabytes. However, if you reach out to me personally by email or through any other channel you prefer, I can just give you a personal link where you can pull this information from through some of these university file sharing systems. So this is very useful. You can just request them from me, and I will happily send them. And as I said, your sysadmin will be very happy because any proper HPC will have some sort of requirement to run Aptena or Singularity files rather than convoluted conda environments. So I'm very happy to share this with you as well. So I now prepared a kind of a code along, a workshop. So if you want, you can do as I do. And since I cannot share my screen, it's even funnier because I'm going to instruct David to do it. So he has to do what I tell him to do, and then we can see if it's really that easy to do. Okay, so next slide, please.
Sorry.
No, all good. Yeah, you're gonna have to use your browser too. Okay, perfect. So now you can end the presentation here. So we're all gonna run some RFT Fusion 3 to figure it out together. So you go on the slide 38 where I shared the GitHub link. Yeah, you click on there. Yeah, or you copy paste it.
It doesn't let me click.
So for those of you, you probably figured out how to.
You can still see my screen?
Yes. Then you go to the structural design, so the second chapter. And in there you click on the Hugging Face. Oh no, further for that, the top one. RFT Fusion 3.
Here?
Yes. Very good. Perfect. Then you scroll down, and at the very last line they write examples. Oh, you went too far. There you go up again.
Yes.
Yeah, you click on examples. The last line. No, further up in the written text, the last line.
Oh, okay. This.
Oh, for you. It's in here exactly. You click on the directory of the repo. You click on the link.
Here. Okay.
You can do it.
Sorry.
Awesome. Well, you pick one, whatever you want, and then you download those. Oh, not yet.
I have to download.
You go one folder out, and then in the middle you see the size, like where it says 6.8 kilobytes, 308 bytes, and there is an arrow in the middle of it.
Yes.
Yes. So you download it, and the other one too, both. Nice. Then you go on the Hugging Face, and to the left you click here. Yeah, you figured it out. Put the JSON there.
Okay.
And the PDB file here. And then before you click run generation, you change the max duration to 200 instead of 300.
Yeah. Can I run?
Yes.
Oh, there's an
What? How did this not work for you? I'll try 150. I don't know. I mean, I conveniently also made slides. 100?
I think I can create a...
No. Maybe that's why. Because I have a Hugging Face account. Could you try 100 again, just out of curiosity? I mean, I put this on the slides, so... Oh well, good.
It's working.
Yeah, let's see. Maybe there's a timeout. Mm-hmm. But I also put it on the slides just in case it doesn't work. So yeah, you can go to the presentation again and let this run in the background. Just don't close the tab.
Yes.
And in there you can go through the results, because that was like, what if something doesn't work? I make it like in a cooking show. So here you can now see that like I used the nucleic acid design, but it's essentially did the exact same thing like the video just did. And then when you get the results, like there's this one little link where you can download the zip file, you unzip it, and then you put it on. I use Molstar, but you can also use PyMol or RCSB, and on there you can then see your first designed protein. In this case, I went for the DNA binding protein, not the enzyme, but in the end, it's exactly the same thing. So if you want to play around with it and just get an idea on how the tool works and the documentation works, it's really good for you to just, like, if you want to design a protein and you have never done this is the best way to start. So for this is really good. And I also use this for all the classes MTAing, when the students are like, oh my God, protein design is so cool, how do we start? I'm like, wait, first you understand it like this, and then we can go on the cluster. So this is great.
We just check if it has finished right. I don't know yet.
Maybe it works. I know for a fact that you will have more resources if you have a Hugging Face account, because I had like a timeout after 250 seconds. And usually for this design, it should not take longer than.
Yeah, no, but it clearly says that it's sufficient to have a free account to access some of the computational power. So.
Yeah, exactly. And Hugging Face doesn't cost you anything. Okay. Well, with this, I'm happily concluding this presentation. And if you have any questions, please go ahead. Thank you.
Okay, I applaud for all the audience since we are online.
Yeah.
I will go back to the slide where you left the link.
Mm-hmm. Yes.
I will actually stop sharing my screen so that, yeah. So first of all, if the audience wants to ask any questions, you can raise your hand and unmute yourself, or if you prefer, you can write them in the chat and I will read them out loud for you. I wanted to start with two questions that are more on the philosophical side, let's say, rather than technical. Mm-hmm. So clearly, these models, machine learning models that you mentioned, are learning something about proteins, how they fold, how they work, and they're also confident whether one design works better than another. And do you think we can kind of learn from what they have learned in their turn, since we are also interested not in just the statistics of generating designs, but also understanding better the processes behind?
I mean, this is kind of a big question, right? When you go into the area of interpretable AI, where you wonder how could this actually, how could it work? And there is people who look into the last layers. They check the weights of the attention operation. How would it look like when you change something in the protein? The problem is it's quite complicated for these structure modules, because they have to operate in a space which makes some sort of mathematical operations. Okay, so these models have to be, call it SE3 equivariant or rotationally invariant. That means if I rotate my training sample by 90 degrees, the protein should realize that this is still the same protein, I just rotated it by 90 degrees. The same goes for the SE3 equivariant, where translation could not change anything in terms of if I move something in the plus X direction, this is just moved by plus X and nothing more. So here it's a bit harder to follow the interpretation, but when you look, for example, at these pure protein language models, where you have only evolutionary data, you can see some sort of pattern in there, where you can see, well, if I would investigate the property of a certain amino acid, then there is really some sort of mask or some sort of attention learned to this specific residue. So I guess the answer to your question is yes, but unfortunately not always.
Yeah, that's still cool. Thank you. I will start reading from the chat some questions. The first one is by Catherine. She asks, are there tools to design proteins that change conformation upon binding? For example, for cooperative binding of multiple ligands?
So there's not really direct tools, but there is ways to get around it. You would, for example, go ahead and use one of these tools I linked before. I talked about conformation is on the way to be solved, where you then provide one sequence and you get you provide two different structures, and this model then tries to find one sequence where these different models converge to. So actually, the answer is yes, this exists. It's just a bit less easy to do, like it's a bit more hands-on, but principle, yes.
Nice. And by Helena, what's the biggest bottleneck when moving from in silico to the lab? What is the type of experimental data we need to make current models more performant?
That's a pretty good question. I feel like I'm not really allowed to answer this question because I only design proteins in silico and never actually go to the lab. But yeah, obviously more data is good. Like when you think of something optimization based or active learning, when you start off with certain design run and you want to improve it based on the data from the wet lab, you would then go ahead and say, well, some of the designs worked not and some of them worked a bit, then you would take those which worked a bit and go ahead and do a new design run. But when you just want to test certain reaction conditions or different properties, you want to optimize for one of those things, I mean, the whole world of be it Bayesian optimization or active learning is open to you. So I feel like the lab is already really good. It's more that how can we combine these two methods in the most efficient way? And I'm pretty sure that Adaptive will mention something like this because most big companies have much more money to go for broader design campaigns and really test their model essentially and these designs, but they can actually also use this data and go back and let this inform the design process as it is.
Interesting. By communist, how much is the success rate of the design? I mean, when you evaluate in the lab? This is the first question. You can start by this.
It goes from 0.5% to 8 to 10%. It really depends on the tool you're using, and it depends on the model system. But as far as I know, that Minecraft or one of the more proprietary models from latent labs, they really report like success rates in the 10% range, assuming that the target is somewhat feasible. So kind of those are the numbers we're dealing with.
And the other question is, can I use these approaches to engineer, let's say, thermostable PTAs, polymerase, or any other enzymes?
Yes, you can certainly do that. Like this is not an absurd thing. We know that proteins which have been designed, like sequence redesign specifically, they are known to be more thermostable.
Then a question by Radu. What on-premise compute infrastructure power would be needed in order to currently work in a productive way in the field of protein design in an educational setting?
Sorry, what was the last word? In what setting?
In an educational setting.
Well, educationally, well, if you don't want to produce a lot of proteins and be like, oh, this is now that working binder on the target I'm interested in, for educational, it's really also fine to stick to the models on the Hugging Face. So, for example, I was allowed to prepare the infrastructure for a hackathon, which we hosted here at EPFL, where we essentially got access to the cluster here, where one part was they had to figure out how to run the models using the things on the Hugging Face, like, for example, this RF Diffusion 3 space I just showed you. But then the other part was to go on the cluster, and on there they then ran these models in a more large-scale environment. But I think if you just want to teach it, like the part where you don't go on the cluster is perfectly fine. I mean, we at EPFL have the possibility to give like access to our educational cluster, which is a bunch of Tesla V100s. And on those, the students can run all the models, not in the large scale as we would do it for our purposes, but everything is normally feasible. So these containers I shared with you, they would run on any environment like this where you have an NVIDIA GPU associated to it, or like CUDA-enabled GPU associated. So I would say anything, maybe something starting with 32 gigs of VRAM would be good, and then the more the better.
It's nice. Yeah, Hugging Face is indeed very open source and education friendly. But let's say, just for the sake of like fantasizing about it, let's say you really care about running it locally, also to lower the costs in the long term. What would be the minimal setup for non-highly productive but just educational indeed
Yeah, I mean, then like the classic gaming GPU, like a more high-end gaming GPU, is probably fine. So if you have a desktop, a workstation standing around with a proper gaming GPU associated, this would be good. Like just for playing around with it, this is more than enough. So consumer-grade gaming GPUs, essentially.
Mm-hmm.
Yeah.
Building on this question also, still again going in the realm of philosophy almost. Do you think in the future there will be more and more specialized labs that will just do protein design on a super powerful computers, or do you see it becoming something that will become more miniaturized and will be just present in every molecular biology lab because it's just too useful?
Well, ideally both, no? I feel like there's different use cases. One of them is where people or like labs like ours or other labs in the community of protein design where they do method development, they also need to train models. You kind of need a cluster for it because it just doesn't work otherwise. Like the protein structures are just too big to fit into small GPU memories. But then on the other hand, when you look at either these services like Tamarind Bio or Latent Labs, or also like what I shared with you, it's kind of much easier to just do it. Like you're a research group, you have no idea about any of this, and you just wanna go ahead and then start like designing something. Then yes, it's quite easy. I mean, you would probably have to put a bit of money on the table, be it to buy one of these workstations or get some credits on Tamarind Bio or one of these other proprietary platforms. But in the end, it will be doable for sure.
Nice. We have one more question by Alonso. He thanks you for the presentation, and he asks, in which area of synthetic biology could be applied the proteins that you are designing?
So in general, I would say anywhere. For me personally, I work on enzyme design. So, well, anything which is related to this. Ideally, like I'm trying to design novel enzymes, and it's a very hard task, but ideally it would be used for biotechnological approaches. But at this point, it's more of an open academic question, like can we design novel enzymes? Not necessarily will it be better than the one which exists in nature, or will it actually work? Yeah, obviously. But yeah, I think in the area of biotechnology, for me personally, I believe that protein design is maybe not so great for like pure pharma, where we have these challenges of biodistribution, immunogenicity. Maybe you go into this antibody specific part, but this is academically not so interesting. But more like in biotechnology. Like for example, you have some toxins flying around outside and you want to design a binder and then put this in an assay, or you have some other more diagnostic things you're interested in where you don't have to worry about, will this now go in the wrong part of the tissue? Like you're making your life a bit easier, essentially. But in the end, it's still a lot of really cool applications to work on, I think.
Nice. Okay. There are no more questions. I just have two small announcements, let's say. The first one is to really encourage everybody from the audience to check out Benedikt's GitHub. There is a lot of work put behind, and there is a lot of stuff around on the internet of what you can use, platforms you can exploit for running online your designs. But this GitHub page is super useful because it kind of summarizes a lot of the tools that are available. It's a super good starting point. And in the end, I think that it allows for each of us to have the barrier being just how much time and effort we want to put, because the resources are already pointed out. So check it out. And the second announcement is that...
Sorry, to interrupt you again. But I feel like if you check this Fusion 3 Hugging Space out and you're like, oh, I have my protein. If you don't know how to make this input file to JSON or YAML or whatever to make it work, you know how to use any sort of large language model and just prompt it and give it the input file so you can try around with it yourself. Like, this is not complicated. Sorry.
Yeah. And the second announcement is about the second session that we have today that was supposed to start at 5. Due to the technical problems in the beginning, we delayed it by 15 minutes. It will feature adaptive bio. And yeah, So we will disconnect from this session and start the next one in a few minutes. And yeah, hope to see you there soon. And thanks again, Benedikt. It was super interesting.
Very happy to.
See you around.
Bye-bye.